Thank you. So the last speaker in the session is Birney, and he's going to talk to us about the ENCODE project. He's going to be brought in on the screen now. So, okay. Can you hear us?
Yes, I can indeed.
Okay, great. You're on.
Okay, and is my PowerPoint screen
Yes,
also on the other side?
Yes, we can see your PowerPoint.
Brilliant. Okay. Well, I'm really sorry that I couldn't be with you in person. I've got an important thing tomorrow, so I hope this PowerPoint presentation is useful. And I'm very happy to answer questions. And of course, people want to contact me as well. I'm happy for that in addition. So let me just go to just the first couple of slides. So slide 2 just shows the situation in 2001. I was one of the people involved in the sequencing and analysis of the human genome, more on the analysis end. And after that, we had two big kind of themes in large-scale data gathering in genomics. And one was about variation and about disease association to variation. But the other one was about understanding how the genome worked. And that was, in particular, the non-protein coding part of the genome. And that's a project called ENCODE. If you go to the next slide, slide 3, you can see that last year we published a big set of papers in Nature. We are sort of 10 years in to a 15-year project. And in fact, rather thankfully, I'm no longer associated in such a high level because it's quite exhausting, as I might come on to mention. But so we're now, that publication is complete. And in the next phase, people are looking to scale what's happened in this publication across the entire genome. But I know that you didn't ask me here to really talk about ENCODE in terms of necessarily the science. So in slide 4, what I'm hoping to show, it's quite hard to work out whether you're on slide 4, but hopefully it's working. It's worth taking a step back and asking yourself, why are we doing large-scale data gathering and why are we doing large-scale data analysis? And the reason why is to provide systematic data sets. And it's very important to realize that the rationale here is usually to allow a broad set of scientists to make discoveries. And so the endpoint here is about a kind of an enabling process for many other scientists to do interesting work or to do work faster or quicker, and in particular to have kind of these serendipitous discoveries where they do something that they weren't thinking about beforehand because the data is so easy and accessible. So just to emphasize that point, on slide 5, I've just given some things which are different between a kind of smaller scale approach and a larger scale approach. And for me, it's very important to realize that although there are tensions between these approaches, I have always discovered that it's when those tensions are translated in a productive way, that's where I find the best science occurs. And it's very important to understand what one does in one modality versus another modality. It would be very dangerous to think that the only science we should do is very large-scale science. But on the same sense, I think the large-scale science has something very different to offer than the collection of small-scale science. Right, so if we go on to slide 6, the final kind of meta point here, before I jump back into ENCODE, is that large-scale data gathering and analysis is because you're trying to enable many other scientists to make insights. There's not much point in doing it unless you can provide full open access of the data and the analysis results. And when I observe other groups in biology and further away, sometimes they don't do this bit. And this bit is very simple. It's about ensuring that all of your data is available and your analysis, but it's absolutely critical to enable other people, of course, to build on it. And that's what the whole point of doing it is. So now I'm going to go more into some pragmatic details. And again, I'm going to sort of take you from a general, and then I'm going into the specific of ENCODE. So on slide 7, which I hope we're on, it's hard for me to tell. Can somebody nod if we're on slide 7? It should say, what are the steps in large-scale data gathering? And you can see here that there's a series of steps: generate, aggregate, organize, transform, analyze. And what I've put beneath them are a series of adjectives. And those adjectives are the things that I think are most important in those different areas. And you can see some adjectives are the same, for example, detail, but there's some adjectives which are very different, like dedication, scalability. It happens only in some of them, and only in analysis do we really get this creative kind of aspect as well. So on slide 8, I take that same kind of scheme: generate, aggregate, organize, transform, analyze, and I just play it out in this area for ENCODE. And in these cases, there were different funded groups for different sections of this, for the generation, for the aggregation and organization, for the analysis. And there's different timescales as well. The data generation, data collection, and data analysis was sort of the centrally funded collection across by NHGRI, but the science groups can and did come from all over the place. And also, to persist this data beyond this funding scheme, there is the long-term archiving group, and that's actually the institution that I'm part of, the EBI, is long-term archive, analogous to NCBI in America. So you have many different groups. I don't think it's right to think of this as a kind of one group process. That's not a very helpful process. You're going to have a mixture of groups with a mixture of PIs and skill sets working together. Now, before I jumped into ENCODE, I felt that it's very important to put slide 9 up, because I'm sure everybody, when they come into this, will discuss about the importance of metadata. And I just want to share hard-won experience here of 10 years of doing this. There are these two extremes in metadata. You can be very light. That's on the left-hand side of the picture here, where in the extreme case, each group has its own freedom to sort of design its little metadata. Maybe it's more taggy and stuff like that. And then on the opposite, on the right-hand side, you can be very heavy, have a single committee, go through a very structured process for that. The trouble is these two extremes both feel very principled. I have to admit, when you're doing it either way, you do feel you're doing sort of the right thing for different reasons, but the extremes tend not to be useful. The lightweight approach tends not to be useful because at the end of the day, there's not enough coordination for you to do joint analysis. And the heavyweight approach tends not to be useful because it's just not responsive enough. And very often the standard, or the metadata standard that's produced, is so heavy that nobody actually uses it. So you're always in the middle. And then just a word of warning about being in the middle is you don't feel like you're always compromising. And it's taken some time to learn that feeling that I'm compromising between these two things is actually an indication that you're doing something right, but it's actually not that pleasant. Trying to work through those compromises. So I'm now going to jump a little bit into ENCODE. In the next slide, which is animated, so if you head forwards and you just keep clicking through, you'll see that there are these 3 major dimensions. But in fact, formally, each of those dimensions are sort of meta dimensions. Each one of those dimensions is itself a dimension's worth of data. And so it's as if we have a sort of 150 times 182 dimensions on every base, which is a bit of a scary space. And we didn't sample that dimensionality directly. We did a much more structured sampling where we did...
Ask your operator for it. Please consult your directory, call again, or ask your operator.
Okay. Hey, folks, why don't we have a break right after Ewan was going to finish. Maybe we take a break now while we try to reconnect him, and then we'll just move on into the next session, George. Okay, so how about 10 minutes? We'll come back at 20 of.
Gosh, I'm sorry. Hello.
A1. Sorry, it said we had a
Okay.
bad internet
Change
connection.
the plan again. Well, why don't we stick around? We were about to
back.
take a break, but nobody actually got up, really, from their seat. So, okay. Can you hear us all right?
I can. Thank you. Do
Okay.
you know which slide you ended up?
Yep, we're on slide 10.
Okay. So if we go to slide 11, slide 11, just to stress, this was very much a consortium effort. And I was just one, in fact, of the PIs of that consortium. And I know Barbara Wald was talking previously. She was another PI. And if you go on to the click one further in the animation, you'll see that there's a number of key people who are the key postdocs that did the data production. And I think these people are very often the people who are easily forgotten in these sorts of large consortium. And if you remember those adjectives, I think the key word here is dedication for them. And it's very important to realize there's this big skill set that's needed to make this sort of thing work. So on slide 12, I'm now talking about some of the analysis. If you notice at the bottom of the slides, under the title, I listed a number of names. You'll see those names change as I go through these slides. And I just wanted to say that when you get to the analysis stage, there is no real, single recommended way to do analysis. You have to choose the right statistical tool with the right properties for the right task. Very often you have to customize quite carefully what you're doing. And so sometimes people ask me, what is the statistical framework that you use in ENCODE? And there isn't a statistical framework. There is a big collection of different frameworks and different tools. So on slide 13, this is one example framework here. This is a piece of rather hardcore statistics created from the Peter Bickle group. They're, from the Peter Bickle group, and he's a non-parametric statistician, and this is to do with how we did some of the not necessarily raw data processing, but the next stage, the very early pipeline processing in this on slide 13. But slide 14 is a very different process done in Ziping Wang's group, where they modeled the behavior of histone modifications with respect to RNA. And in this case, they used a random forest model. Again, I'm not going to talk you through the details to this, except the fact that this is the kind of quantitative modeling that one might end up doing. In this case, it's a very statistical-based model. And then in slide 15 here, we have some of the work that I enjoyed the most, where we took a very unbiased approach to the data, and in this case used two Bayesian frameworks, a dynamic Bayesian network and a hidden Markov model, to learn features directly from the data where we could compare known data sets to it, and in this case it's known annotation. And if you go on to slide 16, there's a piece of animation between 15 and 16, so you might need to click twice. Slide 16 shows us going back and doing targeted experiments. You notice Barbara Wald is noted in this slide. And in this case, we took some of the hypotheses that we generated and tested them back in the lab. And closing that loop is always a very important thing. Now, in slides 17 and 18, well, I'm just going to give you a sense of what I think is the most important result of ENCODE, which is the fact that we see association of this data with things that are associated with disease. And that's been very informative on slide 18. Again, this is not something that I expect you to necessarily understand, because I mean, it's very understandable. It just takes a much longer time than I have to talk about it. But what this table on the left-hand side is diseases, and across the top are different transcription factors and cell lines. And the green cells are associations of this particular cell line to this particular disease. Now, in many cases, these are things that we've known about previously, which is sort of reassuring, but there's one or two of them that are very, very interesting. And I know many of these are being chased up now in specific disease groups. And it's that result of enabling other scientists to make these discoveries, which is the real reason why you do this. It makes me very happy to see. Slide 19 is just to remind you about this business of generating, aggregating, organizing. And I just want to talk about that aggregating and organizing step first off. And in slide 20, I talk a little bit here, or show a number of cases, ways that we have to produce access to this. And on the bottom right there, you notice that the large-scale archives, or raw data and GeoSRA are there, but then there's also display methods, such as the UCSC Genome Browser, my favorite browser because it comes from our institute, Ensemble, and new things that we made specifically for this data set, such as Factorbook. Now, just to mention, this project, ENCODE, is actually not the biggest data project in the world. In biology, the 1,000 Genomes Project and cancer projects are much bigger than this. But we were quite demanding. I mean, 300 terabytes of space is not tiny, but it's not huge. We needed a reasonable amount of CPU time. Again, I've seen bigger projects. We did need a lot of bandwidth between the different sites so that people could trade big data sets quickly. And as long as you're on the high bandwidth things, I think that works. But it's very important, when you really start pushing bandwidth, you have to understand all the bottlenecks in your system. Very often those are local. But I want to stress that as well as that physical infrastructure, we did need very much an information infrastructure as well. So we needed to have components of knowledge that we could take this large-scale data set and integrated it with. And that's where the longer-term archiving processes and value-added processes are very important. So on slide 23, I just go through some thank yous here. So if you go through the animation, you'll see, so there are 440 authors listed here. Quite obviously, you can't really read those, but I want to pull out two sections. So right at the top, Ian Dunham, who is just a gifted organizer and biologist, and he worked very closely at the EBI with myself. And Anshul Kunjai, who was at Stanford, now at MIT, were two absolutely key people. And then the next group of people, as I mentioned, are the people who often don't get the credit that they should do. And these are the dedicated postdocs behind making high quality data. And it's, you know, what people often want and talk about is the endpoint, the analysis, but without good high quality data, there really is no point in trying to analyze it in a sophisticated way if you don't have good data. And these guys deserve a lot more credit than they get, I think. Now, I was trying to think about it from your perspective, as people who are not necessarily biologists. I don't think you're listening to this because you're interested in the biology. Just to say that there are some things from myself here that you might be interested in reading. And I'm very happy for this PowerPoint distribution, the slide set to be distributed. Alongside ENCODE, I wrote a commentary, and rather than being 440 authors, there's just one author, myself, about ENCODE management. So this is not about the overall, about all components. It's a rather narrow commentary in some ways. But I think that there are some important lessons that we learned through this. And then thinking about the broader thing, I blog as a kind of mini, you know, publication where you don't have to argue with editors, basically. And there are these 4 blogs I think are interesting, or I would think are interesting. And amazingly enough, you can follow me on Twitter. But there's some other resources and projects which if you're interested in how biology does this and think about this, here are some other projects to look at how they're organized and run. I wanted to put a structural component genomics project into the mid-sized project, but I didn't manage to find the right URL. And then if you haven't read this book, I would really recommend The Signal and the Noise from Nate Silver. Worth. I find this book, it's a very nice book, very accessible if you're not computational, but if you are computational, like myself, it's reaffirmed to me why Bayesian models are good and worth reading. So I'd like to leave it there because I know we're running out of time, and I'm extremely happy to answer any questions, assuming we can get the communications to work well. So great.